right, Ann, I think we can get started.
Okay, great. Welcome, everyone, to the DuchenneConnect Direct Access webinar series. My name is Ann Martin, and I am a genetic counselor and the coordinator of the DuchenneConnect Registry. Joining me today as the moderator is Ryan Fisher. Ryan is the Director of Outreach and Advocacy at PPMD. So everyone should be able to see the webinar slides on their screen, as well as the chat box for questions. Please feel free to send questions throughout the webinar, and we will try to address them as we go along or at the end of the webinar. And whatever questions we don't have time to answer today, we will provide you with an answer by email within the next week. So please do feel free to send questions in to us. The title of our webinar today is Updates in Genetic Testing. This webinar is actually a collaborative effort. We are very lucky to have three wonderful speakers joining us today. Our first speaker will be Madhuri Hege. Madhuri is the Senior Director of the Emory Genetics Laboratory in Atlanta. Our second speaker will be Dr. Susan Sparks. Susan is a clinical geneticist and researcher at Carolinas Medical Center in Charlotte. And our third speaker will be Jodi Wolff. Jodi is the Director of Clinical Services at the MDA. Ryan, do you have anything you want to add before I continue?
No, I think that's a good opening. We want to thank everyone for joining us, and we thank our three wonderful speakers. And with that, we can get started with Madhuri.
I have some intro for Madhuri. So, as I said, Madhuri Hege is a molecular geneticist and the Senior Director of the Emory Genetics Laboratory. Her areas of interest include muscular dystrophy and novel and high-throughput techniques to detect sequence variants. Her research is focused on functional analysis of sequence variants in muscular dystrophies and translating what is learned in the basic research laboratory into clinical practice. Madhuri has been involved with the DuchenneConnect Registry for several years, and we're very fortunate to have her as one of our scientific advisors. So, Madhuri, I'll turn it over to you.
Thank you, Ann. Good morning, everyone. What I want to do today is go over some basic genetics for DMD and also talk about the new techniques or new tests which are available now to test for DMD and BMD. These tests have become available in the last 3 to 4 years and have significantly advanced our ability to detect mutations in the DMD gene. So the first slide basically goes over what we are going to talk about today is review the basic genetics, describe the mutations in the dystrophin gene, go over some of the tests which are currently available, including the new test, and look over I have put in a couple of slides which, even though they are technical, will actually demonstrate the advances in the testing that have taken place and also discuss carrier testing. So first, and I'm not able to, okay, sorry. So why are genes important in DMD? Both DMD and BMD, Duchenne and Becker muscular dystrophy, are caused by mutations in the dystrophin gene. And the dystrophin gene actually codes for a protein, the dystrophin protein. In both DMD and BMD, because of the mutations in the dystrophin gene, there is an abnormal protein which could be completely non-functional or have partial function. This slide talks about actually what is a gene. So if you can imagine our chromosomes or our DNA is a part of a book, which is a book of life and which has got several chapters in it. Each chapter then has several pages, and each page has got pieces of information written on the page. So if you can imagine that as what our genome is, it's got several pieces of information separated into pieces or chapters, which we can call chromosomes, and each chromosome or each chapter has got several pieces of information or letters written on each page, which is a gene and which has got specific alphabets written in it. Now each gene has got two important parts to it. It's an exon and an intron. Essentially what it means is that exon contains a message required to make the dystrophin protein. So this little box here on the slide actually shows how the exon and the introns are linked together. Basically we know the letters which actually code for the dystrophin protein, so we call it exons. So exon 1, 2, 3, and then in between are the introns. When the dystrophin protein is made, the introns get removed and the exons are essentially linked together to form the dystrophin protein. The next slide shows where the dystrophin gene is located. It is located on the X chromosome. So therefore we hear this again and again that it is an X-linked inheritance because it is on the X chromosome. And it is the largest gene in the human genome. It essentially has got 2.2 megabases of letters in it and does occupy a significant part of the gene. It's a very big gene. It has got 79 exons and it is probably the third largest gene in terms of number of exons present in the gene. But the interesting thing about this gene is that the introns are big and the exons are tiny. So there are about 200 letters in each exon and more than 50,000 letters in each intron. Then the gene also has certain areas called promoters. What the promoters do is help the gene get started, in making the protein. So these promoters are placed in various parts of the gene, and because this gene is so big, it cannot be functioning the same way in different tissues in the body. So in the brain, in the muscle, in the other areas of the body, this protein can get truncated or be smaller because of the presence of these different promoters which are spread out throughout the gene. Over a period of last 10 years or so, we have realized that there is not one change change of one letter in the gene that causes DMD or BMD. There are several changes which are called mutations which are spread out throughout the gene which cause DMD. For this reason, the laboratory people had to develop techniques which can help us identify mutations throughout the entire gene. So this next slide actually shows what are the different types of mutations that can happen and which is the most common mutation in the dystrophin gene. The interesting thing about this gene is that compared to the other genes in the human genome, the mutation spectrum or the type of changes that happen in this gene are the proportion of the type of changes that take place in this gene are different than the other genes. So shown here in this slide is the gene itself, and one of the type of deletions which actually results in removal of an exon, which is called a deletion, is shown in the box here. So you have exon 1 linked to exon 3 because exon 2 got deleted. The other type of the mutation that can happen is that when the exon actually gets duplicated. So in this picture you can see that exon 2 is shown twice, and that's a duplication mutation. The third type of mutations that can happen in this gene is a point mutation. So if a deletion or a duplication is not detected, then we have to look for a change of a specific letter in the exon region. So again, going back to what we saw on the first slide, on the second slide, where we had the chapters, the pages, and the letters, we are now looking for a change in the specific letter. So the 4 letters in the DNA are the A, T, G, C, which you see here, and a specific change of any letter can lead to a mutation. Now, not all changes can lead to a mutation. That is something which we do in the lab here to determine that when there is a change, is this a mutation? So there are 2 types of mutations which are shown here. In the first one is a nonsense mutation, where the protein stops or cannot be made, and we designate that as an X. The second type shown here is that instead of taking out the entire exon, a smaller part of that the letters is taken out. So here what is shown here is removal of 3 bases or 3 letters A, T, C, and that can lead to a deletion mutation. Also, the ATC can be duplicated or inserted again right next to it, and that is called an insertion mutation. But these types of mutations can lead to a truncation or stop the protein from being made. So the next slide shows what are the different types of mutations, or what is the spread of these mutations for Duchenne dystrophy and Becker dystrophy. So the deletions are the most common mutations, which is 60 to 65% in Duchenne and slightly higher in Becker. The duplications range from 5 to 10%, and our ability to detect duplications has increased over the years with the ability to use the new technology in the testing laboratories. The small mutations or the changes in the specific letters are about 30 to 35% in the Duchenne and 5 to 10% in Becker. Now, one important thing to note is that 2/3 of the mutations are inherited from the mother, who is a carrier. 1/3 of the mutations which are detected in the DMD gene are de novo, meaning that a new mutation actually happened by chance in the mother. So the question is that why is it important to be tested? The benefits of clinical testing is, the first thing is to confirm the diagnosis. Sometimes it is possible that the diagnosis may not be clear, and it is important to document the diagnosis with proper laboratory testing. It is very helpful for the family members because we can do risk assessment and actually give some guidance with respect to the actual disease process. And then the last one is you can get involved in research studies and clinical trials for DMD. There are a number of clinical trials which have come up in the last few years, but they all require that the diagnosis is confirmed by molecular testing. This is important because these trials are based on the type of the mutation that is detected. So testing becomes very important. Now, what are the limitations? The limitations are sometimes multiple tests are required. And the reason behind that is that right now, where we are with the technology, we need to perform at least 2 different tests to detect a deletion and a duplication and a point mutation. The same method cannot, or the same test cannot detect both type of mutations. So for deletion and a duplication test is negative and the clinical diagnosis is DMD, then we need to look at the specific letters in the gene to identify the point mutation, which is done by a different method. It is also possible that we may not identify a mutation. And it is important to understand that if there is a clinical diagnosis, then that stands true even though we have not found a mutation, because it could be the limitation of the test performed. Now, many laboratories use different methods to perform the test, and it could be because of the method that is used, or it could be simply because the type of the mutation is such that we are not able to have a test developed right now which can look at that or detect that kind of change. Sometimes the results are unexpected. The lab geneticists may not be able to interpret the result or understand what this change is actually doing. And we get about 5% of the results in our lab like that. Our goal always is to give as much interpretation as possible when sending the reports back. But every now and then we do end up with results which we cannot interpret. So the question is, what can we do at that time? The laboratory has to take the responsibility to maintain that change which cannot be interpreted in the lab, and we encourage the physician to contact us from time to time to see if we have been able to interpret. Interpretation keeps changing because as information becomes available in the literature. The lab results do not indicate prognosis or severity. That is done in collaboration with the clinician. And sometimes the cost and the time for the results can be longer than expected. The cost essentially depends on what method is used and what type of the payment method is actually used when doing the testing, and I'll talk about that a little later. The time for results. The labs have always been trying to reduce the time from the time the sample comes to the lab to the time the report is issued. And we try and do it within 2 to 3 weeks, but sometimes due to unexpected results, there could be a delay in issuing the report. So the next slide actually shows how the test can be done. Usually the test is done on a blood sample, and it can be sent directly to the testing lab. But it is important to ask questions about what method is used, what is the detection, can this method detect all types of deletions, duplications, cost, whether the lab can do insurance and billing, turnaround time, when the report will be available, and possible results by keeping in contact through the genetic counselor or the physician with the lab. The provider can order the testing directly to the lab, but there are 2 types of testing that can be ordered through the institution and by using the insurance. Many clinical labs in this country today have a pre-authorization process where they have billing specialists which will take the insurance information and make sure that the test can be ordered. So when asking for the test to be ordered, it is important to talk to the physician or the genetic counselor who's helping. So as I said before, following the clinical exam and the CA values, the first tier of testing is to look for deletions and duplications because those are the most common mutations in this gene. And there are different methods, and these are all technical terms: MLPA, array CGH, quantitative PCR, multiplex PCR, and codon. The bottom 2 methods are the most traditional methods and do not have a very high level of sensitivity. The quantitative PCR came around in early 2000 but was quickly replaced by the technique called MLPA, which is right up at the top. And even today there are many laboratories which actually use MLPA. But in the last 4 years or so, we have been able to develop a new method called array CGH. Why is array CGH good? I'll show you a couple of slides about that. But that method has essentially made a significant advancement in our ability to detect mutations in this gene with a higher sensitivity. So again, the genetic testing pathway essentially means that if a deletion duplication is not detected and the clinical diagnosis indicates DMD, then we have to look for changes in those specific letters by looking for those changes by using a method called sequencing, which is a sequence analysis. Within sequencing, many methods can be used, and some of them are listed here, but essentially to look for that change of a single letter, we have to use a method called sequencing. So the acceptable methods currently in the clinical testing arena are the CGH, MLPA, quantitative PCR. Many laboratories have stopped doing multiplex PCR now, but there are some which actually do it, but they are not able to look at the entire gene at the same time. When the mutation is known, you can just go and look at the single mutation without having to do the full test. So if the method Southern blot is performed, it is important to understand that it does not tell you where the deletion actually ends in the dystrophin gene. It can tell you which exons are deleted, but what is the exact break point. Multiplex PCR, not all exons can be tested at the same time. So we developed this method called array CGH. And this is a very simple slide, and I'll go through it a little bit. At the top, you can see exon 1 to 79 of the Duchenne gene, and below it is a case which we tested in the lab. The line along the middle is a normal line where there is nothing happening in the gene. Anything below the line is deleted, above the line is duplicated. So in this case, we found a deletion of the exon 8 to 13, and the mother did not carry the deletion. But interestingly, when we did the same sample on MLPA, what we saw was a different result. I just want to focus on the lower two boxes and this arrow which points to 13, which is telling actually exon 13. This is a normal sample, and you see the peak of exon 13 higher here than in the mother. But our array CGS test was telling us that the mother does not have exon 13 deleted. So when we actually went and looked at that part of the gene and sequenced it to study the letters, we found that the MLPA probe, or these two red and blue bars, were actually causing a false positive result, or we call it a dropout, due to presence of two letters at the same position. So this test was not being very accurate, and the mother actually did not have the deletion of exon 8 to 13 in her blood sample, and it was very likely a new mutation that had occurred in this family. So with this, we now have the ability to do a more advanced and more sensitive test. So the next two slides are just examples of how sensitive this test is. This is a deletion, a larger deletion of exon 17 to 44, then a deletion of 48 to 52, and then easily the duplication of exon 2 to 4. Duplication mutations have been traditionally very difficult to detect because most of these tests are based on visual observation and looking at the intensity of a band which corresponds to those exons. In this case, we can very easily detect duplications now. And since we started doing this test, we have been able to resolve many cases which have been reported negative in the past. Now the question is that can this test equally detect with equal sensitivity in females as well? And this slide actually shows that it's very easy to detect mutations in females as well. So showing again a deletion of 46 to 55, a smaller deletion of 49 to 50, and then a duplication of exons 18 to 38. So the carrier testing of females becomes very important because then you can confirm the mutation in the family. This helps with genetic counseling and family planning, and many reproductive options are now available. If for more information, this information is available with the DuchenneConnect coordinators or the genetic counselors who are looking after the families. With that, I'm going to stop and hand over to Anne again.
Okay. We have had some questions come in. I think we'll try to address just a couple of them before we move on to Susan. One person asked if Athena is the only lab that does testing or if there are other labs out there. So, Madhuri, if you want to comment on that.
Sure. So, Athena is not the only lab doing the testing. I'm at Emory Genetics, and we do the array CGH method here, and we actually have got proper permission through Athena to do the test here. So most of the testing in this country for many, many genes is done quite easily because these genes have not been patented. Unfortunately, for the DMD gene, it was patented and an exclusive license was given to Athena. So the labs who want to do the testing for DMD have to seek a permission from Athena to do the test. And some labs have done it, some labs have not taken the proper permission. We actually have got the proper rights to do the testing here now.
Okay, great. And one other question involves more prognosis. This parent asked if there's any correlation between deletions and duplications and lifespan.
So currently, the way the lab reports are issued is that they only say that whether there's a deletion or duplication. And the reason we do not give that the prognosis on the report is because it's very important to correlate that with the clinical presentation and the progress. There is some things which we can do when we issue the report is whether we can see from looking at the protein whether the number of exons taken out change the reading or the letters too much or will not have such a dramatic effect on the protein. And we do say that in the report. But again, the prognosis will depend on after the physician gets the reports and does a clinical correlation.
Okay, great. Thank you, Madhuri. I think we will move on to our next speaker now. That is Dr. Susan Sparks. Susan is a clinical geneticist at Carolinas Medical Center in Charlotte. Her area of specialty is neuromuscular disease, and she is the clinical geneticist for the MDA clinic in Charlotte. Susan is also a researcher, and she was recently awarded a 5-year NIH grant for drug development in Duchenne. This grant was actually awarded to 3 institutions, to Children's National Medical Center, the University of Pittsburgh, and Carolinas Medical Center. And together they will be the Center of Research Translation. And they will be focusing on expanding opportunities in exon skipping, as well as other compounds that will hopefully be changing the outcome for everyone with Duchenne. And I actually had the pleasure of working with Susan at Carolinas Medical Center, and I'm very excited to have her joining us today. So I'll turn it over to you, Susan.
Well, thank you, Anne. I hope everyone can hear me, and I appreciate the opportunity to talk to you a little bit about the upcoming or therapies that are currently in clinical trials and why genetic testing is so important for the boys with Duchenne. So I'm going to talk about the importance of genetic testing for the treatment of Duchenne and Becker muscular dystrophy, focusing specifically on mutation-based therapies. Hold on, let's see if I can figure out the slides. Okay. So this slide is not a slide to be read thoroughly. What it's there to do is show you that Duchenne was first described back in 1860 and the late 1800s, but it wasn't until 1980s or early 90s when the gene was actually identified. Since that time, we have expanded testing for the gene and have developed specific therapies for Duchenne muscular dystrophy. And I'm going to focus on 2 of those therapies. One is exon skipping, which has been shown to be a potential therapy for patients who have deletions or duplications in the dystrophin gene, and stop codon read-through. And these are for those patients that have the X, those single point mutations that have an X in their gene change. So in order to kind of talk about this, I'm going to go back a little bit about the difference between Duchenne and Becker muscular dystrophy. Patients with Duchenne muscular dystrophy, there is no dystrophin protein produced. These deletions and duplications or the point mutations lead to instructions that do not produce protein. The deletions and duplications are typically what we call out-of-frame mutations, and I'm going to discuss exactly what that means. With Contrary, with Becker muscular dystrophy, some of the dystrophin is produced, but the amount that's produced is decreased or not as functional as normal dystrophin protein. So these patients have a milder course than patients with Duchenne muscular dystrophy. The deletions that are seen within the dystrophin gene are what's called in-frame deletions, and those result in the production of an abnormal but still partially functional dystrophin protein. The point mutations that are seen in Becker muscular dystrophy lead to decreased protein produced, not no protein produced, like in Duchenne. So if we go back to the example that Madhuri used with the words, this kind of explains the reading frame theory. So if we take the original gene to say the old fat man and his old fat dog ran and ran and ran and ran for the bus, and then we look at Duchenne muscular dystrophy, and the X's are what causes those letters are missing. When you put the words back together, you can read the old, but then the rest of the sentence is nonsense, and it doesn't make sense. It doesn't tell us what the sentence reads, and it doesn't produce any instructions for the machinery in the cell to make protein. On the other hand, in Becker muscular dystrophy, if you have a deletion, and it can be larger than the deletion seen in Duchenne muscular dystrophy, but if it's in frame, meaning that you're deleting whole words, the sentence still makes sense. So in this case, the old fat man and his old fat dog ran and ran for the bus is the resultant instructions. This next slide, I think, shows you this with those exons. And the reason I bring this up is this shows what's called the reading frame and the ends. And what you want it to fit together is like a puzzle. In the first example, when you have a deletion of exon 48 to 50, the exons that are left, exon 47 and 51, cannot come together like a normal puzzle. So that instructions are out of frame. However, in the next example, you delete one more exon, specifically exon 51. Now 47 can hook to 52. It fits the puzzle, and instructions are produced to produce functional dystrophin protein, although it's smaller. This is kind of shown here where in the top part where you have a normal dystrophin, and the function of that is to bridge the cytoskeleton within the muscle and keep it intact. The example of the Becker patient just has a truncated or shortened bridge, but it's still functional, whereas with Duchenne, you don't make any protein, it's out of frame, and that bridge function is lost, and you end up with a severe phenotype. So what antisense oligonucleotide therapy is, or exon skipping therapy, these are small molecules that are chemically created DNA of 20 base pairs, and they are sequence specific to target the Duchenne gene at certain areas to skip exons. It's also used to knock down expression, and they've used it in laboratory research for a long time. But more recently, it's been used to force the cellular machinery to skip exons, making an out-of-frame mutation into an in-frame mutation. So here's the example of a patient that has Duchenne muscular dystrophy who has a deletion from exon 48 to exon 50, which is out of frame and does not form the proper puzzle, and you produce no functional dystrophin protein. If you use an antisense oligonucleotide to exon 51 and skip it, now you're deleting from exon 48 to through exon 51, and you restore the reading frame and form a truncated but functional protein. So here's the example again, kind of blown up, showing what was described in the previous example. So here it is back to the words where we used that same example before with the old fat man. If you delete or block that T, it restores that reading frame. So whereas in Duchenne, we can't understand that sentence, if you block the T, which is red in this picture, the result is the old man and his old fat dog ran and ran and ran for the bus. We have restored the reading frame, and we can understand the message to make functional protein. And here's another analogy using an anchor, whereas dystrophin is like an anchor for a boat. The middle picture depicts a Duchenne patient with no functional protein or no anchor, and the last picture shows a shortened anchor that is still functional. Here's the current status of exon skipping. So this therapy began with phase 1 and phase 2 trials, which are the early trials to look at safety back in 2007. It started with skipping of exon 51. We have now had development of molecules that can skip not only exon 51, but also exon 44, 45, and 53. So it's very important to know the breakpoints of the boy's deletion or possibly duplication to see if the molecules would be able to benefit the child. I'm now going to switch over to stop codon read-through and bring up the other form of therapies that can be used for those patients that have point mutations that lead to a stop codon and no functional dystrophin protein being produced. This may be somewhere between 15% and 25 or 30% of patients with Duchenne muscular dystrophy. There has been developed small molecules that actually bind to that cellular machinery and allow that machinery to read through that stop codon, resulting in a functional protein being produced. Now, this is a figure from the PTC Therapeutics and website, which shows the normal machinery in the top figure where the protein is produced, which is that green blob on the right. If there is a stop codon, the machinery cannot read through that, and there's an incomplete protein produced that is not functional, or even no protein produced. The small molecule that can read through that point mutation can allow a functional protein to be produced. And there is still research going on in this area. So we go back to the benefits of genetic testing. Right now, our treatments for Duchenne is symptomatically based. There are treatments available that have prolonged ambulation, that have improved the respiratory and cardiac complications of Duchenne muscular dystrophy, but they are focused on the symptoms of Duchenne muscular dystrophy. These new therapies, such as exon skipping and stop codon read-through, are very mutation-based, and they are individualized for each patient. These therapies do have potentially a large impact on the disease progression, but need to be initiated before the symptoms are too far progressed. And even if there's no interest for patients to participate in clinical research, the specific DMD mutation needs to be determined for medical treatment, because after these molecules go through the rigor of clinical trials and get FDA approved, they will be part of clinical practice for your physician to prescribe. Without knowing your mutation, it's difficult to know which of these therapies would benefit your child. So I'd like to thank you and turn it back over to Anne.
Okay, thank you, Susan. We've had several questions come in that are specific for your child's genetic testing. So I just want to let everyone know that we are happy to review your child's genetic test report. So you can either email it to me. My email is coordinator@DuchenneConnect.org. That's actually, I think, on the last slide of the presentation. Or you can fax the report to me. My fax is 404-986-6666. 935-0636. And I'm happy to review the report and can also discuss it with Madhuri and let you know if your child needs any further testing at all. And we did have another question, Susan, that I think I'll have you address because I think it's a common question. One parent asked, will exon skipping ever be used for duplications?
I think it can be used for particular duplications if skipping another exon bordering that would allow it to go back into frame. So it's going to rely on that. The other possibility, depending on how big the duplication is, so if it's a small duplication, there's the potential that they could block the duplicated portion. But it's going to be a little bit more tricky and more specific for duplications. It's more straightforward for the deletions.
Okay. Okay, great. Thank you, Susan.
Mm-hmm.
All right, we will move on to our last speaker of the day. That is Jodi Wolff. Jodi is the Director of Clinical Services at the MDA, and she is also the Director of the MDA Summer Camp Program. In addition to her role at the MDA, Jodi is currently a doctoral candidate in rehabilitation at the University of Arizona. She studies the transition to adulthood of youth with neuromuscular disease, and she also teaches a disability studies course at the University of Arizona. Today, Jodi will be discussing payment issues surrounding genetic testing, and we're very grateful to have her here today with us. So I will turn it over to Jodi now.
Thanks so much, Anne. Thanks for having me today. My slide is actually not up on the screen, but that's okay.
I'm trying to get that up. There we go.
I can do it without.
Everyone, can you see that now, Jodi?
Yeah, there we go.
Okay, let me move to the next one.
Here we are. Great. So today I just want to briefly touch on paying for genetic testing and also insurance coverage for genetic testing. And so it's really important to have a working knowledge of your insurance coverage and really getting to know and asking the questions about your insurance plan. So it's important when you're going through the diagnostic process or, any new referral process really for new tests. It's important to know ahead of time what your insurance company covers and specifically what procedures are required in order for them to cover that testing. Insurance companies, as you probably all know, like to put up a lot of hoops, be very specific in the procedures and the documentation that is required for them to cover a specific test. Also, get to know what the appeals process is, because often when people get denied by their insurance company, sometimes it's easier just to stop there. But if you know the appeals process and you work with a good social worker, a good genetic counselor, whether they are involved directly as part of your clinic team or not, they can help you appeal any denials from your insurance company by reframing and being very explicit about why that testing is necessary and what the impact that testing will have on treatment as well as the rest of the family and the cost efficientness of that testing. And so, and be sure to, if necessary, if your insurance company is giving you a lot of problems, there are insurance advocacy services that are available. And your local MDA Healthcare Services Coordinator, myself, PPMD, we can all direct you towards insurance advocacy services. And then we can also engage in that as well on your behalf. And so it's very important also when you're going to your MDA clinic or your local neurologist that referrals and pre-approvals are received before appointments. Insurance often requires that you get their pre-approval prior to attending an appointment at an MDA clinic or with your local neurologist, whatever the case may be. Now, if you do not have approval from your insurance company to see that physician, or if your insurance company denies your visit and does not cover what they may consider an out-of-network visit. If that doesn't happen, then any of the services ordered as a result of that visit, so if they send you to a cardiologist or if they run more tests or take blood work, all of that may not be covered because that initial pre-approval was not received. And you could receive a very large bill from your clinic visit. You may also have difficulty getting your insurance to cover genetic testing if the ordering neurologist is not covered by your insurance. So what that means is that your neurologist may refer you back to your primary care physician and coordinate directly with your primary care physician for your PCP to order the genetic testing and the blood work. That may be what your insurance requires you to do. Now, that's not saying that your primary care physician needs to figure that all out. They can send a requisition form for the genetic testing and fill it all out for you and send everything they need to the primary caregiver. That neurologist can still coordinate that, or your genetic counselor. But it may be actually your primary care physician that needs to coordinate that for insurance purposes. Now, it's very important to work with a genetic counselor and social worker, as you've seen from Dr. Hege and Dr. Sparks, that they are the experts in this testing. So it's really important to get connected with a geneticist or a genetic counselor. They can be your advocate. They can tell you everything from where you should seek out testing, how to get insurance coverage for your testing, where you can get testing that it will cost you the least amount. They can instruct you in carrier testing, when to get carrier testing for your daughters, how that gets covered, what your out-of-pocket responsibility is. The geneticists and the genetic counselors can tell you everything from what color tube the blood work's supposed to be in so it's processed right, to how to get the paperwork filled out, to order the testing, and how to appeal that through your insurance if your insurance is saying they're not going to cover it. From what I've seen also is that a number of people that have asked questions on here have talked about, do I need to get my son retested? And if that's the case, there may be insurance issues, but working with a good genetic counselor, they can make sure that it's explained to your insurance company in a way so that your insurance company understands that this is what needs to be done, and the importance and the impact that redoing this testing will have on the care and treatment of your child. It's also important that, not every clinic team that you work with will have a genetic counselor in clinic readily available. So it's important that if your clinic does not have one, that you seek out a referral to a genetic counselor. The vast majority of the clinics have a specific genetic counselor that they make referrals to, someone that's skilled in neuromuscular diseases. And so it's important to seek that person out if they're not made readily available. So there is some self-advocacy involved as well, is to make sure that you're connected with a genetic counselor. All children should have health insurance. There are a lot of resources out there now. So if you're having difficulty getting your child covered for one reason or another, or if that insurance is very expensive, or if your private insurance is making it very difficult to get things covered, work with a social worker so that they can connect you with resources, whether that be through the hospital or state-funded or federally funded programs for insurance, because there may be a number of options out there, particularly for kids with disabilities. I just want to briefly touch on what the MDA clinic, how that functions and what is covered. Because as the Director of Clinical Services, I tend to get a lot of questions about that. Now, MDA provides block grants to about 200 clinics across the country. Those are clinics of varying sizes. Some have very large multidisciplinary clinic teams. Some may be in more rural areas or in areas where some may see 1,200 patients a year. Some may see 50 to 100 patients a year. It just depends on the geographic area. We provide them with block grants. We provide the clinic director with a pool of money. We specify that that with that money, they have to cover a patient's visit with the physician so that we can make neuromuscular care available to all people regardless of their insurance coverage. So you will always have the opportunity to visit with the physician. Now, that does not always cover copays and deductibles because some hospitals, based on state laws, hospitals in different locations may interpret their insurance contracts and laws differently. So in some areas, covering copays and deductibles out of the MDA grant is considered a breach of the insurance contract. So in some areas, you will have to pay for copays and deductibles. But other than that, the physicians have the freedom to use their block grant for how they feel that money best supports their clinical services and their neuromuscular program. So that may be covering specific tests and procedures for families. That may be providing salary support to some of their multidisciplinary team members, like their social worker or their occupational therapist or the dietitian, or other team members that aren't able to bill directly for their services. And that's really going to vary between MDA clinics. And so it's important to work with your healthcare service coordinator ahead of time to find out what will be covered, what your expenses will be going into clinic, what you can expect the MDA block grant will cover, and what it won't. And what we're finding is that more and more clinic teams, because of the financial and economic issues with hospitals right now, they're certainly feeling the crunch as well. And so what we're finding is that in order to maintain those multidisciplinary, strong neuromuscular teams, that they're having to take their block grant money and shift a little bit away from covering diagnostic testing to supporting the time and the space of having their multidisciplinary team there. And that's what we're seeing really across the country. That is also going to vary between clinics based on the amount of support from the institution, from the hospital as well. Now, with all of that being said, there is alternative funding. Sometimes there's alternative funding through hospitals and research grants. If you're having difficulty getting your genetic testing covered through your insurance and you've worked with a genetic counselor and social worker to no avail, there may be funds through the hospital. Sometimes those are made available based on income level. But sometimes hospitals do have funds for families that aren't able to cover out-of-pocket expenses for diagnostic testing. Also, there's a large amount of research going on right now. And because it is vital to that research that every child involved in that research study has a confirmed diagnosis of Duchenne or Becker, and that they know specifically what the deletion, duplication, or point mutation is, that many times they will build funding, financial support into their research grant for genetic testing of children that have not had it. So there is a high likelihood that you could get genetic testing covered through a research, under a research grant, and work with your genetic counselors and as well as, MDA and PPMD to find out where that might be available. And so I am happy to answer any questions at this time, and I will turn it back over to Anne. Thank you so much.
Okay. Thank you, Jodi. Appreciate that. We've had so many questions come in. Unfortunately, we're not going to be able to address them all right now, but like I said, we will do our best to get answers to you, this week at the latest. I did want to mention a couple things. There's been several questions about carrier testing, and one parent asked about using a CK level for carrier testing, and I just want to emphasize that we no longer use CK to test carriers. It's not very accurate. A lot of female carriers can have a CK level in the normal range. So for carrier testing, you really need to have genetic testing done, and the genetic mutation first needs to be found in the affected male in the family. And I think another parent asked if all mothers should have carrier testing. And I would definitely say yes, even if you're not planning to have more children. Finding out if you're a carrier is very important for your own health, because we're now finding that a small percentage of carriers can have cardiac problems. So it is very important to have carrier testing done, not only for family planning issues, but also for your health. And I also wanted to comment on something else. There's been questions. One question in particular asked if the data that goes into the DuchenneConnect registry, if we send that on to treat NMD, and we definitely do. And I just really want to encourage everyone to please send in your genetic test report. We do review them very closely. We let you know if you need additional testing, and all that information is then fed into the Treat NMD, the global DMD registry, and that is being used by researchers to develop new therapies. So the registry is very important, and we really need your information. So everyone, please register and send in your genetic test report. Okay, Brian, did you want to add anything?
No, just a reminder that these slides of the presentation will be posted online, and all the questions will be answered that were submitted, will be made available for sure by end of week. Okay.
Do we have time to address a couple other questions?
Sure, absolutely. Go for it.
Okay. One parent asked a good question, whether or not there are any other therapies for stop codon read-through other than PTC 124 or Adalurin. And I am not aware of any, but Susan, I don't know if you have anything to add, if you're still on.
Yeah, I'm still here. I don't know of any. They had tried antibiotics in the past, like gentamicin, to do stop codon read-through, but it wasn't very efficient at doing so. And they're always looking for those kind of molecules to help, because it not only helps Duchenne muscular dystrophy, but a lot of other genetic conditions. So there is a lot of research being done in that area, but nothing that I've seen come to the extent that PTC 124 or Adalurin has come.
Right. Okay, great. All right, well, I just want to thank all three of our speakers again. You were wonderful, and I want to thank everyone who is listening and participating in the webinar today. And I just want to encourage you to please join us next month as well.
All right, great. Thank you, everybody.
Okay, thank you.
Have a great day.
Thanks.